Hello, biologists. Let's take a look at the Meselson Stahl experiment. So the experiment showed that the DNA replication is semiconservative. What does that mean? So let's draw here a DNA molecule. And there are a couple of models to describe how the DNA replication happens, right? The first one is the conservative model. And the conservative model is like a photocopy. So you have the original molecule and you just make a new molecule of DNA copied from the first one. So why is it called conservative? Because you conserve the first one and then you make a copy. The semi-conservative model, on the other hand, you conserve just half of the old one. So you have half of the double strand is the old strand, and then you have a new one interlaced here, paired with the old one. So you have the old strand and you have the new strand on the same molecule. So the old molecule with both strands being the old one, the red one here, they don't exist anymore. You have one molecule in which it's half red, half blue, and the other one half red, half blue as well. This is the semi-conservative model. And then there's the dispersive model, which is a bit messy because you have parts that are old and parts that are new mixed in the same molecule. So it's something kind of like this, more or less. So parts of the molecule here, double strand new, double strand old, double strand new. It's a bit of a mix. So this is the conservative, this is the semi-conservative, and this is the dispersive. And what Meselson and Stahl did is they showed that this one is the right way. The cells actually do this one and not the others, right? So let's take a look at what they did. First, they took some bacteria and they grew these bacteria in an environment full of nitrogen 15. Now, if you remember, nitrogen normally would have atomic mass 14. So this nitrogen has one additional neutron, which makes it heavier. These bacteria, they grow for many generations in this environment where their only source of nitrogen was this heavy nitrogen here, nitrogen 15. So all their molecules that have nitrogen, they are heavier than they normally would be because the nitrogen is heavy. So what are the two molecules that contain nitrogen? DNA, well, three molecules, DNA, RNA also contain nitrogen, also protein. So what we're going to do, we're going to look at DNA, not the other one, not the other two. So these bacteria, they have heavy DNA because their DNA has this heavy nitrogen. Then they took these bacteria and they put in a medium, in a growth medium, containing regular nitrogen. Regular nitrogen 14, not 15 anymore. So these bacteria are growing here in this environment in which their source of nitrogen is regular nitrogen 14. So they're making DNA that is lighter. So the DNA strands that are being made now, they are lighter. So from that, you can start imagining what is going to happen. They not only stopped here, they continued with their experiment. They transferred these bacteria to another generation, still nitrogen 14. So all their nitrogen here is 14. And these bacteria are making regular DNA, not heavy DNA anymore. And what did they do? They took all these bacteria, extracted their DNA, centrifuged their DNA, And when you centrifuge, you can separate molecules by mass. Right? The heavier ones will move, and then you can separate them in a gel. You can separate them by mass. And this is what they obtained. So here you have Meselson and Stahl having a drink, smoking a cigarette, because scientists are not all, those guys who look like Einstein with their tongues out and white coats. Scientists are also people. They grew bacteria in nitrogen 15 first, and then several generations in nitrogen 14 to see what their DNA would look like. Centrifuged, so you spin real quick to separate them by mass, the molecules by mass, and this is what they got. So the first generation, whoa, what's going on here? All right, so the first generation with the nitrogen 15 here, you can see the band with the DNA, and it has a peak here at this line, The next generation, the second generation has nitrogen 15 and nitrogen 14. And you see that there's another band. It's not the original band, it's one single band that peaks lighter than the original one. So here is heavier and here is lighter. So light, heavy, and you can see that the peak is lighter than the original one. The third generation is even more curious because you have two peaks now, not only one. They're equal peaks. You still have nitrogen 15, but you have more nitrogen 14 because now it's the second generation under nitrogen 14. So more molecules are being made with nitrogen 14, and you have one peak here that is lighter than all of them, and one peak with the same magnitude here, the same as the previous generation. But now we also have the lightest peak so far. The following generation, the following fourth generation, they have two bands. You still have that peak here that is the same as the second generation, the same here as the first generation to get in contact with nitrogen 14, and a stronger peak here at the light, right? So you don't have one extra lighter peak, but instead you have a stronger peak at the lightest here that showed up at the third generation. And if you could go more and more, you would see this peak here increasing and this other one decreasing. Note that you never ever had the first peak when all the DNA was heavy. It never appeared again. The first peak never appeared again. It was gone. After the first generation, it was gone. So let's imagine here. What if the DNA replication was conservative? Well, then you would have nitrogen 15 in the first generation. Then the second generation, you would have the nitrogen 15 and nitrogen 14. So you would have one strong heavy peak and then one peak that was lighter and one peak that was heavy. And the following generation, you would still have the old one, the new one made from this. You would have, let's use a different color for this, right? So it came from this one, and then you would have this and a copy that would come from this one. All of those are made of nitrogen 14 because that's what's available. And you would still have the peak. You would still have the peak here at the nitrogen 15 and a stronger, 3 times stronger peak for nitrogen 14. This is not what we see. This is not what we see at all. See, compare here. This is not what we see. We see the peak of 15, and the second generation, there is no peak of 15. And there's no peak of 14 either. You have an intermediate, almost as if it was a 14 and a half. This is why we know that the DNA replication is not conservative, because it doesn't conserve the peak of nitrogen 15 DNA. It disappears. So DNA replication is not conservative. Well, what is it then? It is semi-conservative. How can we interpret these findings? First, we have the peak for nitrogen 15, heavy DNA. But since you conserve half of the molecule with nitrogen 15, and the other half, it's the new strand made of DNA 14, each of these molecules weight the same. So you will not have 2 bands, you will only have 1 band because they weigh the same. And half of it is made of nitrogen 15, half of it is made of nitrogen 15. So it is an intermediate. So this is 15 plus 14. Both of these, they weigh the same, and they are a mix. They are somewhere in between pure 15 and pure 14. So this is why you have this peak here, because then you have a peak or this intermediate, right? You have this intermediate peak here. And then what happens is this molecule is going to conserve 1 old, create 1 new. This other one here is being conserved, and a newer one will be created. Same thing happens to this side. You have 1 old and 1 newer being created, and this one here, 1 old and 1 newer being created. This lighter blue is still nitrogen 14. Remember that nitrogen 14 is all they have. So out of how many strands? So we have 4 molecules, but how many strands? 8 strands, right? We'll have 1, 2, 3, 4, 5, 6, 7, 8. Out of these 8 strands, 2 are heavy, the rest is light. Out of these molecules here, you have 2 that are still intermediate. I'm going to use a different color. These 2 are still intermediate. So you have the peak of the intermediate, but also 2 of them are light. Both of these strands are made of nitrogen 14, so you have the lighter peak of 14. So you have the peak of 14, the peak that is intermediate, and no peak for the heavy one. The heavy one doesn't show up because you don't have this pure red molecule anymore. Not gonna happen. It's not going to show ever again. These guys replicate. What happens? This red one is conserved. This red one is conserved, but they pair with new strands and they are light. So you still have the intermediate ones. But each one of these 1, 2, 3, 4, 5, and 6 strands, all of them, 1, 2, 3, and 4, and 5, and 6, they all pair with new nitrogen 14 molecules. So you don't have a peak for the heavy one. You have a light, small peak here for the intermediate and a stronger peak for the light. How much stronger? We have 2 intermediates 1, 2, 3, 4, 5, 6. So this one will be 3 times as big as this one. And as the generations progress, the light peak will be stronger and the intermediate peak will be smaller, and you'll never have the heavy peak again. This is how Meselson and Stahl showed that DNA replication is semi-conservative. What does that mean? You conserve one of the strands and the other one is new. The old strand serves as a template for the new one to be made.